Dynein is a family of cytoskeletal motor proteins that move along microtubules. They convert the chemical energy stored in ATP to mechanical work. Dynein transports various cellular cargoes, provides forces and displacements important in mitosis, and drives the beat of eukaryotic cilia and flagella. All of these functions rely on Dynein's ability to move towards the minus end of the microtubules, known as retrograde transport. Thus, they are called minus-end directed motors. The structure shown here, from PDB entry to 3VKH, is the most complete structure so far. The entire motor is composed of one long chain that folds into many functional domains. The core of Dynein is composed of a ring of 6 AAA+ domains, similar to the engines that power the unfolding of proteins in AAA+ proteases. One of these, the AAA1 domain colored dark blue here, is connected to a linker and a long tail, both in magenta. The tail usually connects 2 or 3 Dynein motors together, and attaches to the cargo that will be transported. The AAA4 domain forms a long stalk that carries a small microtubule binding domain, supported by a short strut from the AAA5 domain. To activate Dynein and start moving along the microtubule, it needs to bind to a protein complex called dynactin. Without it, Dynein does not work. As the first step in the mechanochemical cycle of Dynein, ATP binds to Dynein and induces rapid dissociation from the microtubule. Remodeling of the linker extends the search range of the microtubule binding domain along the microtubule. After hydrolysis of ATP to ADP and inorganic phosphate, the motor domain is thought to engage a new binding site on the microtubule, initially via weak interaction. Strong binding to the microtubule accelerates the release of phosphate from AAA1, inducing the linker to revert to its straight form. This transition is speculated to represent the power stroke, the main step in which force is transmitted to the attached object. Finally, ADP is released from AAA1 and the cycle restarts.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues, or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
Life requires energy. The universal biological fuel is a molecule called ATP, or adenosine triphosphate. ATP stores chemical energy in the form of a high-energy phosphate bond. As the name suggests, it consists of an adenosine ring with a chain of 3 phosphate groups attached. The last of these phosphates can be split off to release the energy within the bond, resulting in adenosine diphosphate and free inorganic phosphate. This energy is needed to keep cells alive and is used from processes from muscle contraction to producing the thoughts your brain is having right now. The hydrolysis of ATP can be reversed by the addition of inorganic phosphate to ADP. However, this of course requires energy and is performed by a molecular power generator called ATP synthase. Here we see an atomic model of F1-FO ATP synthase sitting in a membrane. This molecular motor is found in the mitochondria of eukaryotic cells and is responsible for making most of the ATP. Mitochondria, the powerhouses of the cells, turn over around 60 kilograms of ATP every day in an adult person. A long chain of biochemical processes convert the energy stored in food into a proton gradient across the inner mitochondrial membrane. These protons drive a rotation of the turbine within ATP synthase, resulting in the synthesis of ATP. Like all generators, ATP synthase consists of 2 separate motors coupled together. Within the membrane, we have the FO motor, named after the binding of antibiotic oligomycin, and at the top we have the F1 motor from factor 1. The FO motor is a proton-powered motor. It is thought that protons flow through a channel open to just the intermembrane space, where they bind to a ring of protein subunits, rotate 360 degrees, and exit through another channel exposed only to the matrix. The net flow of protons driven by the proton motor force provides the energy for the generation of rotation. The torque generated in the FO motor is transferred to the F1 motor by a central stalk or shaft. The F1 motor is responsible for generating the ATP by addition of phosphate to ADP. The top of the central stalk acts similarly to a camshaft, so that as it rotates within the F1 motor, it causes conformational changes of the catalytic subunits.
What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility and an intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer.
Wait a second. Richard Dawkins thought intelligent design might be a legitimate pursuit?
And that designer could well be a higher intelligence from elsewhere in the universe. Well, but that higher intelligence would itself have had to have come about by some explicable or ultimately explicable process. It couldn't have just jumped into existence spontaneously. That's the point.
So Professor Dawkins was not against intelligent design, just certain types of designers, such as God. So the